In the last video, we discussed mitosis. Mitosis is a process in somatic cells that produces genetically identical daughter cells. So they should be genetically identical to the parent and genetically identical to any sister cells that are produced. In the process of mitosis, you go from one parent cell to two daughter cells. But we need to move on now and talk about meiosis, which is the production of gametes, or the production of eggs and sperm. So this process happens in either the ovary or the testes of the individual, depending on whether or not they're male or female. It is really important in completing the sexual lifestyle cycle of an individual. There are a few differences with mitosis that we will talk about. The two major ones is that in mitosis you start with a parent cell and duplicated DNA just like meiosis. In meiosis you start with one parent cell and duplicated DNA just like in mitosis. But in meiosis you have two cell divisions. Because you have two cell divisions, you end up with 4 daughter cells instead of 2. The purpose of the second cell division is also to reduce the chromosome number by half. So two major differences. Mitosis produces 2 genetically identical daughter cells. Meiosis produces 4 cells with half the number of chromosomes of the parent cell. And as we'll see in a few minutes, each of those daughter cells is genetically different from one another. The process of meiosis is really important in reproduction. So reproduction is one of the characteristics that we said all living things have. So whether you are a bacterium, an archaea, a protist, a plant, a mammal, all of these organisms reproduce. So chapter 10 is where we really get into genetics and the study of inheritance, or passing on genetic information to the next generation. So you'll notice by looking at any family picture that offspring resemble their parents, sometimes quite closely, sometimes not so much. Sometimes they're identical to one parent, or sometimes they're a really good mix of both of them. So offspring resemble their parents, but they're not identical copies. So as we enter the study of the unit of genetics, we are going to be talking about heredity. So heredity is the study of inheritance, or passing on genetic information from one individual to the next, one generation to the next. Genetics is just the study of heredity. It's the study of how genes and genetic information are passed on and how they work in an organism. Because we're talking about producing offspring, we're going to be focusing on gametes, or the sex cells in an organism. Gametes are different from somatic cells in that they have half the number of chromosomes. They are what we call haploid. They have one copy of each type of chromosome. This is in contrast to the somatic cells that have 2 copies of each type of a chromosome. And then because we're entering genetics, we are going to start talking about the concept of a gene, which is a single unit of information in a DNA. It is a short sequence of DNA that codes varies for a specific trait within an organism. So using DNA as sort of the blueprints or the information for the cell is one of our unifying themes in biology. All living organisms use DNA or RNA as its source of genetic information. Passing on DNA, or the study of heredity, begins with DNA replication. So DNA has to be copied before it can be passed on to the next generation. It's sort of like if you give someone an instruction manual, you still need that instruction manual. So you make someone a copy before you give it to them. It's the same idea with DNA. The parent cell has to maintain its copies of DNA, but the daughter cell needs its own copy as well. So we have a few new terms that we need to talk about. And these relate to chromosomes and reproduction. So autosomes are our chromosomes that are not the X and Y chromosomes. The X and Y chromosomes are the sex chromosomes. They help to determine the sex of an organism. In humans, we have 23 types of chromosomes. 22 of those types are autosomes, and then the other are the X and the Y chromosomes. We have 2 copies of all of our autosomes. Remember, that means that we are diploid organisms, so our cells have 2 copies of each type. Those copies, one from mom, one from dad. Our gametes, though, our sex cells, have haploid number of chromosomes. This means they have only 1 copy of each type of chromosome. And the reason for that is once the organism reaches reproductive age and that sperm unites with an egg, fertilization occurs, we get back to diploid. And this is why we have mom giving 1/2 of our chromosomes and dad giving the other half. If the process of meiosis didn't reduce the number of chromosomes, then every time a sperm fertilized an egg, you would double the number of chromosomes. So meiosis has to half the number of chromosomes so that we can get back the right amount at fertilization. Once fertilization occurs, when you have a sperm that is united with an egg and successfully fertilized it, you now have a zygote. So a zygote is just the term for a fertilized egg. If you are a human, that zygote is going to immediately divide and eventually become a fetus. Same thing with other animals as well, but we're mostly going to be discussing humans. Oops. So there are a couple of different types of reproduction that occurs in living organisms. And the 2 types of reproduction are based off whether or not the resulting organisms are identical or different from one another and from the parent. In asexual reproduction, the individuals are genetically identical to the parent and to each other. So a single individual is the sole parent. That means that one parent passes all of the copies of all of the genes into the next generation. So there's no exchange of genetic information. There's no second parent. So we have an example here of something called a hydra. That is a small plant, and it reproduces by budding off. It has a specialized cell there, or group of cells at this branch point here, and they actually reproduce by mitosis and produce an organism that is identical to that parent. All of those offspring that are produced are called clones. A clone is just a group of genetically identical individuals. This is not the same thing as a twin, but you can kind of think of it that way. Redwoods reproduce by asexual reproduction as well. A sort of shoot comes off of one of the roots and grows up next to the parent plant. So all of those trees in that grove of redwoods there are clones as well. They're all genetically identical to one another. The second type of reproduction that we have is sexual reproduction. So sexual reproduction is different in that 2 parents are required to produce an offspring. And the other thing that's important about sexual reproduction is it produces genetically different offspring. So those offspring are different from either one of the parents, and they're different from all other siblings in that generation. This is really important because it allows for variation within a population. So the most important aspect of sexual reproduction is that there's variation that's produced. We will see during meiosis, some variation is produced. And then when you have two parents coming together, bringing unique combinations of genes together in an individual, you get even more variation. And that variation is going to be very important for us when we start talking about evolution. But that variation due to sexual reproduction is important to the organisms for evolution as well. If you have a group of individuals that are all genetically identical to each other and something changes in their environment and they can't respond in a way that can be inherited, they're probably not going to survive. But if some individuals have variation that allows them a better chance of survival, that variation is going to become fixed in the population. It's going to become part of the population. So before we get into meiosis, let's talk about chromosomes just a little bit. So how many DNA molecules are present here? Remember that we said we count chromosomes based on the number of centromeres. So here we have this large blue chromosome. This is considered one chromosome because they're bound together at the centromere. We actually have two molecules of DNA, though, forming one chromosome. So there are a couple different ways we can answer this question. We have 1, 2, 3, 4, 5, 6 pairs of chromatids. So we have 6 sister chromatids. There are actually 12 pieces of DNA, but there is only 6 chromosomes because we're counting them based on their number of centromeres. If you look at the blue and the red large chromosome, these are drawn to indicate that they are the same type with different origins. So one of these chromosomes came from mom and one came from dad. There we have our diploid situation. We have 2 of the same type of chromosome. Same thing with this medium-sized chromosome. This is used to indicate a origin from one parent and the other. Though they're the same types of chromosomes, we have 2 copies of each type, so that is the diploid. So our diploid number here is going to be 2n equals 6. That means there are 6 total chromosomes, but there are 2 copies of each. So our 2n indicates 2 copies, 6 indicates the total number of chromosomes. The haploid number then would be n equals 3. The haploid number tells us that the number of types of chromosomes is 3. So there are 3 distinct types. We have large, medium, and small. Each of those blue ones would have, say, come from mom, and the red maybe came from dad. So here we have our diploid number of 2n equals 6 for 6 total chromosomes, 2 copies of each, and a haploid number of 3. 3 types of chromosomes. If this were to go through meiosis, each of those cells would get 1 large, 1 medium, and 1 small at the end. So we've been talking about sister chromatids. Those are chromosomes that were produced by DNA replication, should be identical, and are attached at the centromere. But we also need to talk about homologous chromosomes. Homologous chromosomes are the pairs of matching chromosomes. So we have 1 chromosome from mom, 1 chromosome from dad. Those are homologous chromosomes. So homologous chromosomes are 2 chromosomes that have the same length. They have the same banding pattern, and I'll talk about that in a minute, and the same centromere position. So basically, they have the same anatomy and they have the same genetic information all the way along. That genetic information can differ a little bit. You can have two different types of genes between mom and dad. Say maybe dad has passed on a brown-eyed gene and mom has passed on a blue-eyed gene, but they're all going to be located at the same place on the same type of chromosome. So here we have our condensed chromosomes. These are chromosomes that are getting ready to go through metaphase, so the most condensed chromosomes. We have our sister chromatids here, so each of these has been replicated from a parent chromosome and is still attached at the centromere. This is showing chromosome 1. So here we have an example of the different types of chromosomes. Chromosome 1 here is the longest chromosome with 2 long arms. And one of these would have come from mom. The other homologous chromosome, or the homologue, would have come from dad. So this picture here is called a karyotype, and it's just a picture of metaphase chromosomes. So cells are taken from an individual during mitosis when they're most condensed, and then a computer software is used to sort of line them up. But if you look at them, you can see that they each have, with their homologue, the same anatomy and the same banding pattern. That banding pattern is produced from different types of stains that stain the DNA at specific places. So each of these homologous chromosomes is going to have the same banding pattern. Sister chromatids should be identical in every way, all the way top to bottom. Homologous chromosomes are going to be the same length, have the same banding pattern, same centromere position, just like sister chromatids do. The difference is that some of those genes that are encoded in the different homologues might be slightly different. So let's move on to actually getting into gamete production and the process of meiosis. So gametes are the only cells that are not produced by mitosis. Remember, we said that all of the other cells in our body divide by mitosis. Parents produce identical daughters. But gametes are a little bit different. Gametes develop in the ovaries or the testes, depending on whether or not you're producing a sperm or an egg. And then, again, an important feature of meiosis is that it reduces sets of chromosomes in half, and that is to prepare for fertilization. So let's look at the sexual lifestyle. I keep saying lifestyle, I mean cycle, sexual life cycle. So in this diagram here, we have the ovary producing the eggs and the testes producing the sperm. That occurs via the process of meiosis. In that process, in the ovaries, eventually we produce the egg. So this is the egg here waiting to be fertilized by this sperm. Each one of those is haploid, which means it has one copy of each type of chromosome. When fertilization occurs and that sperm meets with the egg, it becomes a diploid organism. So now we have one copy of each set of chromosomes from mom, one copy of each set from dad, come together to get your full diploid set. This is where we get our total number of chromosomes for humans is 46. So in humans, the diploid number is 46. That means we have 2 copies of each type of chromosome. Our haploid number is 23. That means our gametes have 1 copy of each type of chromosome. So here you can see the distinction between the haploid stages and the diploid stage. In humans, the haploid stage is actually a very quick, very short part of the life cycle. That's not true with other organisms, but in humans, the only point that an organism, quote unquote, is haploid is when they are a sperm or an egg. Once fertilization occurs, they become diploid, and then the rest of their life, the individual spends being a diploid organism. This bar should actually be on the other side of these people because they're diploid. All of their cells are diploid. but that's neither here nor there at this minute.